Welcome to New Intrinsically Enteric Capsule Technology for Pharmaceutical Drug Development, presented by American Pharmaceutical Review and sponsored by Capsugel. My name is Mike Auerbach, Editor-in-Chief of American Pharmaceutical Review, and I will be the moderator today. Before we begin, I'd like to inform our viewers that this event will hold a live question and answer session at the end of the presentations. You can submit a question at any time using the Q&A box on your screen. Also, please note the resource box containing information and documents pertaining to this webinar. And now, allow me to introduce today's presenters. A biochemist by training at the Bordeaux Polytechnic Institute, Frédéric Bordes-Picard also holds a Master of Business Administration degree from Kedge Business School. Frédéric has been working in the pharmaceutical industry for more than 15 years, first at AstraZeneca UK, working on analytical development of therapeutic proteins and antibodies, then at the French CRO Bertin Pharma, working on generic developments. Frédéric joined Capsugel in 2010 as a pharmaceutical business development manager, providing technical and regulatory support for new capsule-based product development, especially for DPI products. Frédéric has now joined the marketing team and holds the global lead responsibility for Capsugel offerings in the inhalation space. Dr. Eduardo Jewell joined Capsugel in 2004 as part of the newly formed pharmaceutical business development team based in Bornem, Belgium, where he supported the expansion of the Colmar and Strasbourg, France-based product development operations. In 2010, he relocated to Boston, Massachusetts, and returned to research and development, supervising lipid-based formulation development from feasibility to scale-up and manufacturing. Ed and his team are currently responsible for business development in areas ranging from oral bioavailability enhancements to modified and targeted release and other emerging segments such as abuse deterrence, pediatric applications, and drug delivery to the lung. Prior to joining Capsugel, Ed worked for drug delivery firm Nanocarrier in Tokyo, Japan, where he was involved in nanotechnology-based drug delivery licensing operations, technology acquisition, and pharmaceutical development programs with the industry, academia, and the National Cancer Center. He received his doctoral degree in materials science engineering from the University of Tokyo, where he worked on the design and use of polymeric nanoparticles for cancer and gene therapies. Welcome, Frédéric and Ed.
Thank you, Mike. I'll start first by providing a few words on Capsugel. Our global footprint gives us an industry-leading supply capability for our core hard capsule products, including customized capsule offering. In addition, our integrated product design, development, and manufacturing sites offer customers an end-to-end partner for the production of innovative finished dosage products. We own and operate 13 manufacturing sites in 9 countries across the globe, producing 200 billion hard capsules and approximately 3 billion finished dosage units a year for more than our 4,000 customers. Just to name a few sites in North America, our Bend site in Oregon is looking at finished product design, development, and manufacturing, and is actually our Center of Excellence for spray-dry dispersion technology. Quakertown in Pennsylvania is looking at product in intermediate development and manufacturing, and actually is our micronization Center of Excellence. In Tampa, Florida, and the team are looking at developing and manufacturing clinical batches as well as ensuring packaging and distribution, and it's also our Center of Excellence for microdosing and powdering capsule. In Europe, I briefly mentioned the Scottish site in Edinburgh, looking at product design, development, and manufacturing, especially liquid-filled hard capsules, and Ploermel site, with our Center of Excellence for soft gelatin capsules, but also the site for newly introduced technologies such as microdosing and spray drying. As every compound and development program is different, we focus on product design and provide a tailored approach to meet our customer-specific target product profile and commercial objectives. We are building on some key technology platforms, as well as on extensive experience in formulating and advancing 1,000 compounds addressing challenges such as low bioavailability, targeted release, or specialized applications such as pediatric formulation or inhalation delivery. Looking at bioavailability announcement, we have an unmatched suite of technologies ranging from spray drying, micronization, lipid-based formulation, and nanotechnologies, and a robust and data-driven technology selection approach. Targeted release, whether it is modified, enteric, or sustained release, is another strong expertise of Capsugel, building on both formulation and encapsulation technologies. This is the topic of today's webinar, looking at intrinsically enteric capsule technologies. This technology is recently distinguished at the CPHI Innovation Awards. With that, I hand over to you, Eduardo.
Thanks, Mike. Thanks, Frederick. Let's continue on then and talk about enteric delivery, some of the basic concepts or foundations behind that, some of the limitations on current approaches to achieve the same. So why the need for enteric protection? Well, basically, the second an API disintegrates, a dosage form disintegrates and releases the API through dissolution in the gastric environment, it still needs to undergo gastric emptying into the small intestine before it's actually absorbed. So for compounds that are sensitive to that acidic environment, we're talking about pH between 1 and 3, depending on the conditions, but to those compounds that are sensitive to enzymes present in the stomach as well, and inversely for those compounds that can generate irritation or even toxicity in the gastric environment, and last but not least, for those APIs that require, that have a very narrow absorption window, for example, that require very site-specific delivery, enteric targeted release, and in this case specifically enteric release, can be of interest. And this applies obviously for small molecules, but also and especially for biomolecules, for peptides, proteins, or oral vaccines that need to undergo that gastric emptying, that are very sensitive not just to the gastric environment, but to some extent, and we'll talk about that in a second, to the heat that can be generated as a result of the enteric coating process. Obviously, site-specific and enteric delivery can be of interest. And more recently, there's renewed emphasis on the delivery of live organisms, that is bacteria or even viruses. There's a quite an exciting field of research around microbiomes, which are obviously also excellent candidates for this type of delivery. Which delivery, the enteric protection or the delivery to the upper gastrointestinal tract has been historically achieved through, again, the use of functional coatings, which are obviously very well established technologies. They're routine, they're again very well understood and extremely cost effective. They still pose a number of challenges in that the coating process is a multi-step coating that can add to the weight of the dosage form, can obviously involve a number of different steps. And there's additional considerations that need to be taken into account, especially when thinking about the target site or substrate of application, in the sense that if, in the case, say, of a tablet, if the tablet is too soft, well, the use of coatings or the application of coatings can lead to erosion. If it's too hard, that can also lead to poor adhesion. And so there's a nice balance that needs to be achieved when applying that coating to make sure we have homogeneous distribution and drying across the dosage form, and especially sufficient protection at the edges, with a number of defaults that can arise, and there's a number of them from capping to chipping, picking, and logo bridging that are nicely described through these pictures here. So thinking about the fact that 90% of early stage drug development is conducted in capsules by the simplicity they're associated to, right? I mean, it's just about opening the capsule, filling, dosing, filling, closing, and dosing. So a very straightforward process. We thought about what could be the implications, and especially what could be the benefits of working with a capsule that's intrinsically enteric. That is, a capsule that doesn't require any further processing, that doesn't require any coating, but that is in itself enteric. And so looking at the different stages of development as early as formulation and process development. Now, of course, having to identify the right polymer, the right mixture, the right formulation for that enteric application can add to the overall early stage concept identification and validation. And there's more likely than not the need for a number of iterations before the actual vivo or expected or desired vivo performance is achieved. There's risk as you move up in scale, of course, of that process or that scale up to have an impact on the performance. And so that there is a number of steps that are required to further validate and to establish the robustness already in early stage formulation and process development. Now, of course, as you move down into scale up and validation, again, there's need to validate a number of different steps. Of course, the need for establishing a robust DOE space around the process itself, and a number of studies associated to the overall scale up that will be required before you go into that first bio study. And again, we're not saying that this is not well known, well understood, and well established. All we're saying is that obviously relying on that functional coatings may add to the development timelines. And so thinking of that, right, avoiding that enteric coating process can reduce the time, the complexity, some of the risk associated in different stages. There's a number of advantages we have thought about here. Some of my favorite really revolve around simplifying the selection of the enteric coating composition, enabling especially rapid screening and optimization of enteric performance. Again, very valuable in the early stages as you're trying to reach that first studying humans, that POC data you're looking for, and obviously down the road as the product and the clinical program matures, there's the obvious advantage of obviating some of that process development, scale up, and validation. So again, just thinking about what I said at first, working with two-piece capsule with capsules, right, and having a product that is, again, intrinsically enteric, without the need for additional coatings, so that can provide not just that protection from the gastric environment, but also from the heat associated to some of the coating steps, is an obvious asset. And thinking about the savings, here's some more benefits that can be thought about in terms of throughout the development. We asked H2 Pharma to conduct a study on the potential savings, since we're talking about timeline savings here, and they identified, the feedback was that potentially up to 9 months could be saved through phase 1 and 14 months through phase 3 by eliminating the need for enteric coating. And as we all know, of course, every day we miss from the market, right? This is not just the money that is spent, that is required in invested in clinical development, but is also patients waiting for and consumers waiting for additional treatments. So again, obviously, tremendous savings here. Then looking now into what Capsugel has come up with over the years, going back to that concept of intrinsically enteric capsule technologies. We have a long and very strong history in capsule development. The first patents on two-piece capsule manufacture through pin dipping into a gelatin solution go back to the 1880s. And those were back in the days of our grandmother company, Park Davis, which later evolved into Warner Lambert, Pfizer, and now KKR. So we have been in the business of making capsules for quite a long time, but we continue to innovate even in that space. And along the gelatin Coney snap, which is the golden standard reference for the industry, right? Gelatin classic. Gelatin capsule that provides immediate release, gelatin being a phenomenal polymer in that it is self-gelling and that provides those beautiful disintegration properties that lead to immediate release. There's a number of things we characterize and we obviously look into when thinking about alternatives to gelatin. There's a number of reasons why a specific program would not want to go into gelatin, and those could be simple, cultural, could be religious considerations, could be dietary restrictions, there's a number of reasons why we could look into the option of developing non-gelatin or alternative polymers. And the BSE crisis, actually 96, triggered our coming up with the first HPMC capsule in our range, but it was 10 years ago that we came up with, that we developed the VCaps Plus. This is a capsule that is truly HPMC and water and nothing more than that. So we actually got rid of the addition of gelling agents, which we have explored in different capsule products. But in the case of VCaps Plus, now we came with an HPMC capsule that is truly independent. That's the disintegration profile of these capsules is truly pH independent. That is, regardless of the pH, regardless of the ionic force of the environment, VCaps Plus capsules provide immediate release, are expected to behave pretty much the same across the board. And then we decided to continue exploring HPMC as a polymer, very robust, very well known to the pharmaceutical industry, has been around for a while. And first we came up with the DR caps. What we did here is we looked at our HPMC capsules, the VCaps, the traditional VCaps, and now we turbocharged them with gelling agents so that the disintegration of the capsules was substantially delayed so that we could provide delayed release. These do not comply with specifications of the relevant pharmacopoeia, but they do provide with a delayed release, which has been a very popular asset, very, very popular benefit in the health and nutrition space for some ingredients that can generate reflux, for example, that can lead to burping effects. These are very popular. This is a very popular solution. But now coming back to the pharma space, we're looking at into enteric HPMC. We have looked at a number of properties, and obviously we have developed two concepts we'll be talking about today, the VCaps enteric and then the intrinsic, that do provide that pH-dependent profile in compliance now with the different pharmacopoeia, the USB and the EP. And there's a number of other things we have looked at, from the oxygen permeability, especially the possibility of capsulating non-solid forms. The very first thing we look at when evaluating a new formulation is studying or understanding the compatibility between that formulation and the shell. As there will be exchanges of moisture in particular, we want to make sure that we understand that interaction between, again, a formulation of the shell and the environment, and that interaction doesn't lead to loss of properties, loss of the functionality, because really the tremendous added value we bring here to the table is not just coming up with a new capsule. This is adding a new level of complexity, that is the complexity of functionality. Now we have capsules that are, if you may think about it, capsules are a very simple drug delivery system, a simple form of drug delivery. Now we're adding functionality by adding this delayed and more specifically this enteric release. And so the VCaps Enteric, the first concept we'll be talking about today is looks like and acts like a two-piece capsule. It's manufactured with pharmaceutical grades of HPMC and HPMC-AS. Does provide, again, immediate and full disintegration at pH 6.8. We have talked about the materials, we have talked about the compliance, and in as much as possible, whenever we develop new capsules, we sort of tell the short story. That is, we developed a new capsule, but there's much work that has gone into every single new capsule product we come up with. One of the differences of Capsugel is even, again, in the spaces we have been active for a long time, we continue to innovate. We have a very active, very prolific research and development group focusing on polymeric science that looks at exploring new alternatives, new materials, and we have explored several thousand. I often get the question, why do you only have gelatin and HPMC capsules? Well, we have looked at thousands of different polymers, but obviously we need to come up with, to identify polymers that are, on the one hand, machinable from our perspective, in the sense that we, as much as possible, would like to manufacture them, those capsules using existing equipment train we feel comfortable with, thinking about scaling up, of course, and supplying your products with commercial scale volumes, but also thinking about you, the end users, in the sense that we want to make sure you're able to fill these capsules in existing equipment. There's no need to reinvent the wheel, while obviously doing this within affordable spaces. So there's a number of considerations that are taken into account, and that's the reason why we cite here the water content and the storage recommendations, in that these will be pretty much or substantially similar to what you're used to, especially looking at storage conditions. Every single carton box that leaves our factories is marked with a 15 to 25 degrees C and 35 to 65% relative humidity. In this case, we have added aluminum type packaging just to make sure that we preserve the functionality of these capsules. Now, talking about the disintegration test performance, the very first thing we did was to, basically take a picture and let the picture speak for 1,000 words. This is what VCaps Enteric look after 2 hours at pH 1.2. These capsules were not sealed or banded, and you can see that at pH 1.2 the capsules, they've been through 2 hours in simulated gastric environments, but see that they have preserved their integrity. You cannot see the contents. The capsules have obviously not opened and or released their payloads. And then when looking at the dissolution performance, or in other words, the vitro performance, the disintegration profile as seen through the eyes of dissolution, in this case of acetaminophen, a very highly soluble drug that would obviously, if there's anything that would, if the capsule were to open, you would obviously see very rapidly release of the APAP in the environment, regardless. And so what we did is to look at the dissolution and see that at pH 1.2 after 2 hours, there's no more than 10%, and that's the compendial specification. No unit with more than 10% release after 2 hours at pH 1.2, and then subsequently very rapid release at 6.8, where it's expected. We expected an 80% at 30 minutes. And then we added some stability data that we continue to generate, and we have more stability data to show that this performance is consistent, is not affected on storage, be that in open or closed containers. In order to further test or evaluate the functionality of these capsules, we looked at comparing them to products that do provide either enteric delivery or site-specific delivery. The first product we compared it to was Tefidera, a Biogen drug. This is DMF that's used for the treatment of sclerosis. And in this case, the dosage form is a gelatin capsule that contains enterically coated microtablets as DMF has been associated with flushing and stomach aches, and also as the formulation is designed to bypass releasing the stomach and the gastric environment and have the DMF released in the intestine when it's actually rapidly metabolized into the active ingredients. And so what we did was to take DMF and fill it directly in VCaps Enteric. We actually also grinded some microtablets and filled them in VCaps Enteric and compare that to the performance of the reference product of the Fedora, and see that from a dissolution perspective. The dissolution of DMF is pretty much or substantially similar across the board, but especially at in the acidic environment, see that we have in compliance with pharmacopeal requirements or specifications, we have no more than 10% release after 2 hours. So in this case, the product was, or VCaps Enteric was evidenced or was shown to be a potential alternative for microtablets in capsules. In another example, we looked at Entocort. This is budesonide from AstraZeneca, and corticosteroids are usually used to treat Crohn's disease. In this case, the gelatin, this is another gelatin capsule that contains coated microgranules, and release of the API is sought after in the ileum and ascending colon, very site-specific delivery. We followed the same approach. We filled VCaps Enteric directly with budesonide with the pure API, compared the performance in the acidic and buffer stages, and as expected, we met the specifications as described before. In one last example, we took Dultalax, a laxative that's used to treat constipation by stimulating local motility in the colon. Same approach here, same sort of data. So very compelling from an in vitro perspective, looking at, again, the disintegration properties, but also looking at the dissolution profile of a number of different compounds. We see that this is a robust alternative to traditional or more established forms of enteric coating and targeted release. Now, when looking at the vivo behavior of VCaps Enteric, we conducted an MRI study in healthy volunteers just to understand specifically the location, not just the location, but especially the transit times and the location of disintegration of the capsules. We were able to confirm, well, the gastro-resistance of the capsules on the one hand. We were able to locate the site of disintegration as the ileum, and with no evidence for influence of gastric residence time and intestinal disintegration times. And the very good news is that the variability associated to the time to disintegration, or obviously, or overall the performance of the product, was comparable in this case for that of a coated enteric tablet. Now, in some other instances, of course, not having more than 10% release is not acceptable for certain type of applications for the reasons we've mentioned before, be that because the compound is extremely sensitive to that gastric environment, to the enzymes present in the stomach, because the viability of certain live microorganisms cannot be compromised, of course. We have developed the intrinsic drug delivery technology, which is now a platform. It also looks like a two-piece capsule, with the difference that the intrinsic does not allow any release in the gastric environment. There's zero tolerance here, and this is what we've demonstrated through a case study here that I'll talk about in a second. But this is, again, another two-piece capsule that is also manufactured with pharmaceutical grades of cellulosic materials. Contains fully approved enteric polymers. It's compliant with all relevant monographs, and as in in our style, I would say, we have developed all of the documentation. There's a lot of work that goes, we have mentioned, into development, into polymer science, into developing new concepts that are tested, validated, subjected to vitro testing, vivo testing. But there's also a very strong regulatory component that goes into work, and that is generating all the documentation that will be used to support your filings, not just in terms of the COA, but the technical reference file, a very comprehensive document that speaks to all of the properties and characteristics of capsules, but into the DMFs that will be used, again, within the context of a filing to support your product development. And then we obviously generate stability data to substantiate the shelf life of these products. In this case, we have added certain very specific conditions at 5 and especially -20, as we know that these capsules are currently being explored for some applications that require extreme cold condition storage. In order to put the Intrinsic platform to the test, we compared its performance to that of Nexium. Nexium, which, as a reminder, are gelatin capsules filled with enteric-coated pellets of esomeprazole magnesium trihydrate, or EMT. This is not just a well-established, but a very successful, extremely robust product. But another reason why we looked at Nexium is because EMT, if and when in acidic environments, turns a bright yellow, which makes it a very visual probe, I would say. Even a few minutes at pH 1.2 will turn the solution yellow, which is, again, a great marker for vitro testing. But so what we did here is we put the Nexium to the test in vitro first, and you see that the capsule obviously is intact at T0, opens as expected within a few minutes, and within 2 hours, of course, the gelatin capsule is gone, but not the enterically coated pellets. They do preserve their integrity, and they do protect, of course, the EMT from release. But as soon as you move them into buffer stage, the very rapid release, all in compliance with the no unit less than 80% requirement. See that the EMT is so sensitive that it actually starts degrading shortly after that. But in a nutshell, in the acidic stage, very quick dissolution of the gelatin capsules, no dissolution of the pellets, all in compliance with specifications. Same thing in the buffer stage. We have complete dissolution of the pellets with an EMT, 94% of EMT released and dissolved after 30 minutes. Now, to challenge the Intrinsic platform, what we did here is we filled it with uncoated pellets, knowing that the capsule itself is not enterically coated, but it's intrinsically enteric. We just filled it with uncoated pellets and put it to the test, really. And so the beauty of the approach is, you see the picture at 2 hours, pH 1.2, the capsule is intact, and were you to slice this capsule in two, you would see that the pellets are still white. There's no yellowing, there's no brown, there's not enough penetration, right, to trigger that EMT degradation. The EMT is not just released, but it's also protected. So very limited permeability to the extent that we have a 0% release here. And as soon as we move into the buffer stage, within 5 minutes you have capsule opening and almost complete disintegration. After 15 minutes, the capsule, as well as the pellets, of course, are gone. So really very strong performance, provides very effective protection of EMT in this case, while meeting requirements from the USB monograph and for esomeprazole delayed release capsules. Looking at now vivo data, substantially similar approach. We conducted a gamma scintigraphy study in healthy volunteers in the fasted state, and we conducted a PK study comparing these two concepts. And the images are there to show that there's actually no release in the gastric environment, but rapid release as early as a duodenum into the small intestine, so great performance there. Something that's unique and I think is definitely worth pointing is that obviously most, in this case, out of 12 individuals, 11 showed side opening, capsule opening within the small intestine. This was confirmed, which tells us that there's 0% release before the intended site of target. That is, there's 0% and there's no release of the payload in the esophagus or the stomach, whereas we found even some in one instance we found pellets in the esophagus, even though they are coated and they do protect the active. In this case, we have guarantee that there's no release of the payload before the intended site of action of target. So this is a quite unique feature of this product. And then when looking at the PK profile, we see substantially similar performance, very rapid onset, slight differences, I would say, in the Cmax, but overall equivalent AUC, so definitely comparable performance in terms of drug absorption in the fasted state, especially the Tmax, which is something that's obviously very valuable here. Now, going into conclusions before we head into the Q&A session, we have then developed two new concepts, what we think are unique, the VCaps Enteric and the intrinsic drug delivery. Both of them are compliant with relevant pharmacopoeias. They're easy to implement solutions, obviously, in the sense that we're talking about opening, filling, closing, and dosing. The VCaps Enteric is a really easy solution for gastric irritant actives or to provide that delayed release. This is a ready-to-use capsule that is, again, compliant with the specifications on a more than 10% release in gastric environments, whereas the intrinsic now provides full gastroprotection, that is 0% release in the gastric environment, so great protection, especially for novel therapies. We're thinking of the peptides, proteins, live microbiomes, for example. A great enabling technology for oral delivery of not just acid, but also heat-sensitive molecules. And this platform, this technology today is available under license. In terms of competitive advantage, well, there are several. I think we have illustrated most of them throughout the talk. These are rapid tools for the advancement of new chemical entities, also of biopharmaceuticals. Easy enough to implement solution in the early stages of development. They eliminate coding process complexity, and there's potential savings, in this case, up to 9 months up to phase 1, and we said 14 months up to phase 3. There's also potential benefits in the sense of there's potential product differentiation for generic 505(b)(2) or even the consumer healthcare space, in that this is an efficient solution for tailored delayed or modified release profile, which can be used in combination, for example, with multi-particulate approaches, and this can also be a powerful brand to differentiate the product and potentially even in some spaces to circumvent certain IP claims. But overall, what we have simplified and streamlined the development process. Now we have manufacturing efficiency that can be achieved by reducing the number of steps associated to the dosage form development and manufacture. So there's reduction of process complexity. This is definitely falls into the realm of quality by design. And the good news is, as we said before, one of the things we very carefully look into when developing new products is making sure that they are not just manufacturable and scalable from our perspective, but can also be used in high-speed filling environments, continuous manufacturing, all of the tools and all of the equipment lines you have in place, also by providing Six Sigma quality levels. With that, I think this is my last slide. I will revert back to Fred and Mike for any questions you may have.
Thanks, Ed and Frederic, for a great presentation. As Ed mentioned, we'll now begin the question and answer portion of the webinar. I'd like to remind you that you can submit questions for our presenters using the Q&A box on your screen, and we'll answer as many questions as possible. So let's take a look now at our first question. And the question is, what will be the weight variability of these empty enteric capsules from the mean/target weight? This could be extremely useful to see if we can fill low-dose drugs accurately if you have an empty capsule weight, if you can have the empty capsule weight well in control.
So this is a very good question, and we are in the process. We continue to improve the weight, to reduce, in this case, weight variability. The good news is that these capsules are manufactured through a well-known process at Capsugel, and as such, we are able to contain the weight variability within acceptable pharmacopeia levels.
Okay, thanks, Ed. Let's move on to our next question. And the question is, what is the disintegration time of the enteric capsule shell in a higher pH buffer?
Thanks, Ravi. That's a very good question. We have looked at different pH, starting as low as 4 and into higher pH, and in most instances we have. As for the data we have shown here, very rapid disintegration, i.e. roughly 80% within 30 minutes, with certain variability, of course, depending on the pH, but within very rapid disintegration performance. Okay,
thanks again, Ed. Let's move on to our next question, which is, have any alcohol dose dumping studies been performed using enteric capsules? If so, can you share the data on their behavior in 5 to 40% weight / volume alcohol in dissolution medium?
Not to my knowledge, but Fred, maybe you want to comment on this?
Yes, of course. A very interesting question indeed, and that would be another nice application for such VCaps enteric or enteric technologies. There has been some testing, although we choose not to include everything in the presentation today, that indeed highlight some benefits into preventing alcohol dose dumping, especially in conjunction with some multi-particulate formulation designed for sustained delivery. So I would say stay tuned, because there will be more on the topic as we are progressing and fully launching this product.
Okay, thanks, Frederic, for that answer. Our next question is, how is the scalability of enteric HPMC capsules compared with others? Are there any flow issues with VCaps compared with gelatin?
So in terms of the scalability of manufacture, this is the same equipment train that is used to manufacture VCaps Plus, which is sold in the billions today, so it's a very robust process. In terms of the machinability at the filling stage, we have conducted some large-scale filling trials and have concluded that there should be no major machinability challenges. Within the limited data we have today, there's no reason to expect there's any anticipation of any challenges at that scale.
Okay, thanks, Ed. Let's move on to our next question, which is, how is the integrity of intrinsic caps maintained in an acid medium without any banding or sealing?
That's a really, really great question, and it's something we're really excited about, because as the capsules go in that gastric environment, they actually swell so that they auto-lock, if you will. As the capsules swell, they become tighter so that there's no need for the sealing or banding. And we have done a number of, all of the vitro studies we showed was conducted without any additional sealing on the capsule. So really great performance.
Okay, and then the follow-up question for that one, are the intrinsic capsules ready for commercial supply?
They are, as we said, this is a technology that's available under license, and we have a number of partners that are currently evaluating them in the clinic, so we're really excited about that. We have conducted, similarly to VCaps Enteric, trials to manufacture these capsules in larger scales, and we have no concerns whatsoever today. Now, of course, we don't expect these tend to be smaller volume type of products, so we don't anticipate the need to run at extremely high speeds in full-blown commercial environments, I would say, or large scale. But the trials we have conducted today show us that, again, no challenges should be expected. As part of the service we provide with this technology, we offer free on-site support for any of your filling trials. We would send somebody to support in any way to make sure that manufacturing process goes smoothly.
Okay, thanks again, Ed. Let's move on to another question. Do you know if the use of surfactants, approximately 40%, could affect the intrinsic capsules filled with lipid formulations?
Fred, you want to take that? A sip of water here.
Okay, so I guess there are two points in the question. First of all, the compatibility of the Enteric capsule technology with some semi-solid or liquid-filled formulation. In this case, I would advise to go step by step and evaluate the compatibility, although we have already some first data showing that the VCaps Enteric especially could be used in combination with semi-solid or even liquid-filled, liquid formulation with an adequate banding on fusion process afterwards. Regarding the addition of surfactants, I don't believe we have, I don't recall any specific data, so again, that would need to be checked.
Thanks, Frederic. Let's move on to our next question, and it is, what is the difference in polymers used for VCaps, Enteric, and Intrinsic capsules?
They are similar polymers. What we have changed or played with is the composition of the polymers and of the mixtures thereof, and that's about all we can say.
Okay, thanks, Ed. Our next question is, can we use these capsules for moisture-sensitive drugs before reaching the target site?
I think this goes back to Fred's comments before. As with any new formulation evaluation, we would recommend conducting compatibility just to make sure that the drug is in this case, the drug is not affected by the encapsulation process. But overall, HPMC-based capsules tend to have lower moisture levels than gelatin, say, and as such will provide better protection for moisture-sensitive drugs. Depending on how sensitive they are, of course, they can have an impact on the shell, and which in turn can have an impact on performance, but that remains, in our strong recommendation is always to first conduct compatibility.
Okay, thanks, Ed. Let's move on to our next question. Rather long one. Let's see. For esomeprazole, although the overall performance between Nexium and the Intrinsic formulation is similar, the Cmax seems to be outside the acceptable limits. What would be the approach to achieve the ratio for Cmax within BE limits? Hope that makes sense.
It's a good question. I think that in this case, we might even suspect that the Intrinsic performed better than the enteric coated granules. But if my memory serves me right, I think we were still within the realm of bioequivalence. Though I would need to get back to Jasmina on that. But it's a very good question.
Okay. Thanks again, Ed. Let's move on to our next question, which is, what is the mechanism with which heat-sensitive drugs remain stable?
It's basically the lack of use of there's no heat involved in the encapsulation process compared to that of coating in certain instances. So that's basically the mechanism. There's no heat. Here we're just encapsulating, and so as such there's no generation of heat, which is a great asset for heat-sensitive drugs.
Okay, thank you. Move on to our next question. What condition was the transit time studied before? Let me start that again. What condition was the transit time study performed, fasting or fed?
This is all fasting data. We have not conducted fed studies yet.
All right, and then there's a similar question. Have you conducted any imaging studies in the fed state or dissolution studies in bio-relevant media mimicking the fed state with the Intrinsic Capsule or otherwise with VCap Enteric?
Yeah, that's work in process. We have still not conducted studies in the fed state.
Let's move on to our next question about VCaps. Are these VCap Enteric solid sold directly to customers based on the order, or do we need a license as in the case of Intrinsic capsules?
Oh, you want to take that?
Yeah, sure. So as Ed was mentioning, unlike the intrinsic drug delivery technologies that do indeed require a license, the VCaps Enteric will be sold following the same business model as for our other two pieces capsules. Saying this as we are still quite early in the launch phase, we are today working very much on a lead user type of approach, but this is perfectly fine to contact your Capsugel representative and ask for a preliminary sample for test, and you will be contacted by one of our pharmaceutical business development manager to actually put the paper in place, in this case a confidentiality agreement, in order for you to receive the sample to test.
Thanks, Frederique. Just want to remind our viewers that if you have any questions, please submit them in the Ask a Question box, and we'll get to them as soon as possible. So with that, let's move on to our next question. Have any of these solutions been evaluated for microbiome formulations?
Yeah, as a matter of fact, they have been, especially the Intrinsic Platform is very popular in that space, where again, we're talking about a guarantee of 0% release, payload release in the gastric environment, which is obviously a huge asset for viability. And so we have a number of partners that are actually exploring this platform also in the clinic.
Thanks, Ed. Let's move on to our next question. The question is, a few years ago, Capsugel also launched Caps Capsules. Could you clarify how this capsule fits into the portfolio of delayed release solutions?
So this is another great question. The Caps was, to some extent, the first foray in the modified release space. And what we did then was to, again, just work with gel and agent in the composition of the capsule to achieve that delayed release. This approach is today very popular in the health and nutrition space. And why there and not in the pharma is because while we do provide that delayed release, we're not strictly in compliance with compendial specifications.
Thanks again, Ed. Our next question is it possible to request a sample of VCaps Enteric capsules for testing? And if so, what is the process?
Yeah, Frederick covered this before.
Yeah.
Go ahead, Frederick.
No, I guess as mentioned before, as you were saying, Ed, please contact your Capsugel representative, and he will walk you through the process that is quite simple to execute a confidentiality agreement, and then being able to get some advice from our pharmaceutical business development team, also to help you or guide to decide what best solution would work for your specific compound or product profile.
Okay, thanks, Frederic. Let's move on to our next question. Have you put the Enteric capsules through the so-called dynamic or bicarbonate dissolution test, as advocated by Physiosolution, Werner Whitecheese, or UCL, Abdul Basit?
Not to my knowledge. I would need to check with our R&D colleagues, see if this is work in process, but not to the extent of my knowledge.
Okay. And I think we have time for one more question today. And the question is, what is the qualitative difference in polymers used for Enteric caps versus intrinsic capsules?
Yeah, I think we covered that, and we basically worked on grades and composition, but that's about all we can say.
Right. Okay. Well, I think that's about all the time we have today. Any questions that we were not able to address here live will be answered by our presenters via email. Also, when the webinar ends, there will be a brief survey that will open in your browser. We would appreciate your responses to the survey. We'd like to thank Frederic and Ed for sharing their knowledge with us, and also offer a special thank you to Capsugel for sponsoring today's event. Please keep a lookout for an email containing a link to view this webinar on demand. Thank you, and have a great day.